A little after 10, so we're just going to get started. There's more people coming that are running a little late today, so we'll just get going here. Just to give you a heads up, you know, we like questions, comments during the meeting, and don't want to scare anybody, but we have somebody here recording this. We've had requests for people that cannot be here to be able to view the meeting and the information that we go through. So we will do our best to keep this moving today. We're going to be covering a wide variety of things today. We'll have a little movie that we'll be watching. We've got a couple. Well, we had two speakers that we were going to have come in. One is not here because of illness, and we just assumed he didn't bring anything here. So we'll try and get through it ourselves, but we've got a variety of different things, some management tools that we wanted to bring to you guys' attention so that you could use it maybe in your operations. We're looking today at a lot of things that affect what you're doing out there, what's affecting it, why it's affecting it, and what we can do to solve some of these problems. You guys have had a lot of information in the past, and I know you get some of the same stuff over and over, and I apologize for some of that, but most of the people tell me that the repetition seems to help sometimes. But today we're going to try and bring some of this stuff all together, help you understand what's going on in your soil out there and what we can do to help alleviate some of these problems going forward. And the goal is with Soil First and Terra Transitional Solutions, which is what I represent, is to help you improve your bottom line. Increasing yields is what everybody is after. The problem being is there are things tied with increasing yields, and increasing yields usually requires increased inputs. And so your costs go up along with everything else, and if you don't get an increase in yield, you're going behind. And I've shown a graph in the past that shows how farm income has gone up over the last years. But the problem is the net returns have gone this way. The net returns have not gone along with farm income. And because of what we've created in the soil, it is like what is happening to us as a population. We're getting sicker and sicker. Part of it is because, and doctors that are truthful and honest say that 90% of disease is nutrition related. And what we're going to cover today is about that in a roundabout way, because what's happening in your soil's not deficient in most things. It's just being tied up. And that's what we're going to look at today. What's tying it up, why, and with some of the technology we're now using, and with with one of the presenters today, we can start to unlock some of those issues so that you don't have to keep pouring on stuff, because you know how it goes when you start taking medication. They all have side effects. So that side effect, when you have a side effect from a drug, then you got to take something else and something else. And this is what's happening in our soils, whether we like it or not, and it's not going away, and it's been building up year after year after year. So those are the things, and that's why we, can you guys see this? Is it a little hard to see? Is that better? Yeah. Okay. I've given you guys, and I like to do this, as you guys know, I've given you guys all the same PowerPoint slides that are up here are in your packets. And it's a little easier sometimes to go through those, because sometimes it's a little hard to see this stuff on the screen. But there's one thing, on the left-hand side, in the back, there's this little couple-page deal. And I'd like you to take a look at that before we get started here. If you'll pull that out. There's 2 sheets with that, okay? So the first one looks like this. The pictures of grass. You should have 2 of them, hopefully. Everybody find them? Everybody have 2 of them? Okay, so if you look here, this was an email sent to us by a guy that was doing his grass, and he had a hydraulic hose blow on him, and he explains things. You can see where it was here, and I kind of there were 3 or 4 pictures he sent, and I didn't want to run all those off. It takes a lot of ink. So I drew the line across where he had it, but this was his lawn. And as he explains, on May 10 he had a hydraulic break. On June 13 was when he took this picture. July 9 was when he took another one, but he applied less than a pint of Biomentum on this area, and he did it in 3 separate times, July 10, 13, and 16. And you guys know what happens when you get petroleum products on grass, right? Kills it, and it stays dead for a long, long time. So the last picture I have there was taken on September 12, and it's the second one there. And because oil and gasoline, petroleum products are carbon-based, and because these little critters love carbon, because this is all carbon and this is nitrogen, when he sprayed it on there, what they did is they used the petroleum product, whether it's gas or oil, they used it for food and cleaned it up. Okay? And I'm not sure how big a spill he had there, how heavy it was, but anyway, I just wanted to point that out to you guys because it's what we're talking about today is carbon again, and as we've talked about so many times before. So anyway,
less than a pint each time or total?
No, total.
Oh, really?
He had less than a pint of Biomentum, and he split that up into three applications, and just sprayed it on there with one of these little quart jars on the end of a garden hose,
Oh, okay.
He just sprayed some on there. So it's the just to give you an idea of the power of what we're dealing with as far as the biological is concerned. So I did put something else in here for you to read and think about, and it's the first sheet on the left-hand side. And everybody talks about calcium, the importance of calcium and silica. And you'll probably have to read this 2-3 times for it to sink in. But it's extremely important, and it's what we're going to be talking about today in some of the stuff we go over. So, okay, with that, we'll get moving here. Do this right. Okay, so this is what we want to do, is wake up your soil, because 90% of the biology in your soil is dormant and not doing anything. That doesn't come from me, that comes from research that's been done. So that's what we're going to talk about today, is waking up your soil. So this is a big issue, and did we put that in here? Yeah. I also included a testimonial, and then should I ditch nitrogen for biologicals? That's a big topic of discussion right now, is can you or should you do that? So this is from a magazine, and so I wanted to just put that in there for you guys to read. I'm not advocating that necessarily, but there's 4 different crop consultants that gave their opinion on this. So just read it. They're not necessarily in favor of doing that, and we'll talk a little bit more about that as we go along here, but they're just for your reading. And then the other one is a testimonial from a producer down in Iowa, and if you'll pull that out a second, I would just like to go through that a minute. But this young man has been using the Biomentum product for a couple years now, and his dad's been using it longer than that. And he just explains how when he was tilling this fall, how much easier it was on his dad's ground right next to it than it was on this ground. So he's just trying to express how he was impressed with the texture and the looseness of the soil, and that's what we're after. Okay. All right. Everybody's talking about reducing inputs. How many of you guys are going to cut down on your fertilizer this year because of crop prices? That's a big topic of discussion, isn't it? And there's a lot of decisions to be made there. And that's kind of the topic of what we're going to be talking about today. How many of you guys are familiar with Ken Ferry? You get Farm Journal publication at all? I get it digitally. And so this guy's talking about, is it possible to reduce your requirements? Now, I've got a guy down in Nebraska. He tells me he has cut back to almost nothing on his phosphorus and potassium, and they've fertilized dry P&K every year, as long as they've been farming, probably. And this year he put a quart of Biomentum on, and he said where he did that without putting any P&K on was the highest yield he ever had on that piece of ground, and he surpassed his dad's yield on corn, who continued to put on the P&K. So there's lots of examples of that. I'm not gonna say that's what you should do, but we're going to look at some of those examples today. So this is a good one. If you guys could go to that slide, kind of keep up with me if you would. So what these agronomists are talking about is, can you cut nutrition, on your crops? And I think the biggest takeaway here is, with good soil pH, farmers can improve crop yields, nutrient uptake, weed control, and herbicide persistence, okay? And that's exactly what we're going to talk about today, how you can do that. Okay, so this is a chart, if you want to go to the next slide. This is a chart, 6 secrets to 80 bushel beans. How many of you have raised 80 bushel soybeans? Okay, a couple back there. Those guys are from Nebraska, by the way. Okay, and they irrigate too. But so the first limiting factor, as we've always talked about, is always 30 bushels for weather, right? You don't get rain, it don't matter. Genetics, they figure 30 bushels. Row spacing, 7. Foliar protection, 6. Fertility, 5. And seed treatment, 2 bushels. So these are what they feel the dream team. Teamwork makes the dream team work for soybeans. But the first thing is always weather, of course. It doesn't matter with that. So this is what we're going to talk about today, just like we've been talking about before, fungal to bacteria balances. So again, as I mentioned before, soils are not deficient in minerals, they're deficient in microbes, and that's what we're trying to get at. Now, do you guys remember this slide from December? We had I showed the one video of this NRCS guy. No, was it the NRCS? Yeah, NRCS guy. And he was talking about biologicals and the need for, that every time we went across the field with a side, there was no fungal left when he tested. But he also brought this slide up with some research, and I'm still trying to wrap my head around this because it doesn't make any sense, but their hypothesis is, and they've done this through testing, that nitrogen, phosphorus, potassium, and organic matter do not have a bearing on plant growth directly. Does that make sense? It doesn't, does it? Because that's not what we've been taught, right? You got to have those things. And not saying you don't have to have those things, but what they're saying is, like here, when they put that much nitrogen on, there wasn't that much difference in growth. When they put all that phosphorus on, no difference in growth. With high organic matter, it didn't matter. High potassium, same thing, right? But when they found out, when you add fungal to bacteria, which is what we're talking about and have been talking about for the last couple of years now, you get, with low fungal to bacterial activity, you get slow growth, but when that goes way up, and this is 0.4 up here, means that's about 40% fungal. And this is 45%. What is the ideal ratio? What did we talk about before? 50-50. Right. You guys were listening. Good deal. So this is what they're talking about here, and this is what we're testing, and this is why we use this little thing here, the microbiometer tester, because we can test all that. It's 90 or 95% as accurate as a lab, to determine how many of these versus how many of these. These are what? What does this represent? Anybody remember? This is the bacteria. in your soil. What percentage in your soil is bacterial? 90%. When it was all prairie, it was 50-50. This is where we need to get to get more of these and less of these. We need to balance that thing out. And we'll show you that today. Okay, this is just an example, this slide here. This is 0.31, 0.1, 1.72. This represents about 50-50 versus about 0.3 fungal in these soils. And that's what makes a difference, and that's why we use the ABC. And you remember what the ABC was? The amino acids, Biomentum, and carbon, right? So that's why we use that or recommend that in furrow, because the Biomentum is 25% fungal. And that is what we are measuring with this, and that's what we'll look at a little bit more today. So carbon is king, you guys remember that. But fungal is a pot of gold, and that's what we're talking about here. And you've seen this slide before. So 10 to 15% of the phosphorus you apply is taken up by plants every year. 80 to 90% is immobilized in the soil and not directly available to plants. And that's going to be the topic of discussion today. So if you've applied enough phosphorus, if you applied phosphorus for the last 10 years, you've only used 10% of it, and you got enough for the next 90 years. And that's no kidding.
Verlyn?
Yes.
What if you haven't applied that much in the last 10 years?
Well,
They say it's still in soil.
it is. And I think I mentioned this last time too, about Ray Ward when he was asked how much phosphorus he has in his soil, and he said 25 parts per million, and the presenter said, no, how much do you have? And he said, let me do some figuring. He said, I have enough in there for the next 1,000 years without adding anything else. But it's tied up. And today, you will understand why it is tied up before you leave here. Okay, we're going to play this video. I wanted to play this early so that you guys wouldn't fall asleep on me. And so I'm going to have Brittany start this thing up. We may stop this as we go along to discuss some things. Go ahead.
I'm Harley Smith, and this is the Master Growers course. Today we're going to be taking a closer look at microbial inoculants and compost teas. Plants and microorganisms have co-evolved on the planet, not just to help each other survive, but to help each other thrive. They've worked together in a mutually beneficial way as they've evolved on the planet. Plants are autotrophic. That means they're self-feeders. They make their own food. Plants take in carbon dioxide through the leaves, they take up water through the roots, and then in the presence of full spectrum light, they do photosynthesis. They make carbohydrates, photosynthates. Some of those carbohydrates they use for energy to grow and reproduce. Some they store in the flowers, in the fruits, in the seeds, in the roots. But some they leak from the roots to feed the plant growth promoting rhizobacteria and the mycorrhizal fungi right in the rhizosphere. And in exchange for those carbohydrates, the microorganisms make biologically active molecules that aid with the uptake of water and minerals. And those are what we call the biostimulants. But today we're going to be looking at the biostimulant factories. We're going to be looking at the superstars of the rhizosphere. And the first time in the history of agriculture, we're able to harvest microorganisms from the most fertile soils from around the world, harvest other microorganisms from disease-suppressive soils, and then combine them in a way that they work with each other. They work with each other instead of compete with one another. We can do things that have never been done in the history of agriculture. We can find ways now, there are microbial blends coming out of the universities. They're able to cut fertilizer usage in half, reduce the nitrogen fertilizer, but increase quality and yield. We found ways to lower, reduce, or even eliminate the need for pesticides and fungicides. And we've learned how to improve quality and improve yield. So this is the new frontier, and it's very important for the future of agriculture. So today we're going to look at some of those superstars from the rhizosphere. Let's start with the mycorrhizae, the mycorrhizal fungi. Myco means fungus, rhizae means root. So mycorrhizae literally means fungus roots. They're beneficial fungi that infect the roots in a beneficial way. They penetrate the root cells, and then as the mycelium, the threads, grow in the rhizosphere, they harvest and mine nutrients. They unlock phosphorus from the soil, they feed it to the plant. So mycorrhizal fungi will increase the surface area of the root zone that plants have by hundreds or even thousands of times. That improves the uptake of water, improves the uptake of nutrients, makes them more drought resistant, and even stimulates root growth. Remember too, the more efficiently plants take up water and minerals, the more chemical reactions they can do, the higher the Brix. Brix is the measurement of the sugar content in the sap. So one of the goals of all of the Master Growers classes is to grow high Brix plants. If we have more sugar production by the plant, there's more stored energy that can be released in abundance of fruit and flowers. There's more stored energy that can be used to fight off pests and disease or to adapt themselves to environmental stress. And there's more stored energy that can be used to take up essential elements for plant growth and reproduction. So growing high Brix plants is the key, but mycorrhizal fungi aid with the production of the sugars by the plant. But one of the main benefits of mycorrhizal fungi is that they unlock phosphorus from the soil. Now phosphorus gets locked up in the soil very easily. It locks up with calcium to make calcium phosphate. That's what your bones are made out of. 95% water insoluble. And if it's insoluble, it's unavailable to the plant. Also, phosphorus locks up with iron to make iron phosphate. It makes the iron unavailable to the plant. So, if there's a phosphorus deficiency in infertile soil, plants will exude signal molecules to attract the mycorrhizal fungi. It infects the roots, and as the mycelium grows through the root zone, it exudes organic acids, it exudes enzymes that unlock phosphorus from the soil, unlock phosphorus from organic matter, and feed it to the plant. So that's one of your first choices you have to make as a grower. Am I going to be doing all organics, or am I going to do mineral nutrition? If you're going to do organics with rock phosphate and bone meal, then you definitely want to inoculate the roots. Very first thing, right at transplant, with the mycorrhizal fungi. It'll aid with the uptake of the phosphorus. Now the downside is it costs an energy budget for the plant to feed the fungus. In other words, over 20 or to 30% of the energy of photosynthesis is leaked from the roots just to feed the mycorrhizal fungi. So, if there's plenty of water-soluble phosphorus, the plants will treat the mycorrhizal fungi as a pathogen. Instead of attracting them to the roots, they'll make enzymes to repel the mycorrhizal fungi. So, if you're growing with all mineral nutrition, the mycorrhizal fungi won't have nearly the benefits as it would in organic fertilization. So mycorrhizal fungi are especially important with organic nutrition or in a starter fertilizer to help unlock phosphorus from the soil and feed it to the microorganisms. Now, the next on our list are the plant growth promoting rhizobacteria. Unlike the fungi, they don't spread out in the root zone, they colonize the surface of the roots. And the plant growth promoting rhizobacteria are the biostimulant factories. They feed on the waste materials from the plants, from the carbohydrate exudates and amino acids, and they change them into biostimulants. Microorganisms will actually make growth hormones, IAA, right on the surface of the roots. Now, why would a microorganism want to stimulate more roots from the plant? That's because the genetic code in every living cell and every living thing says the same thing. Be fruitful and multiply. So if the microorganisms, if the bacteria stimulate root growth, there's more surface area for the microorganisms to colonize. Also, if there's more surface area, there'll be more leaked carbohydrates to feed the microbes. So it's a competitive advantage for the microorganisms to stimulate root growth. Microorganisms such as the plant growth promoting rhizobacteria also produce B vitamins. Now, a high dose of B1 right on the roots stimulates cellular metabolism of the microorganism themselves and the other microorganisms in the root zone. And at the same time, the B vitamins stimulate the systemic induced response of the plant. It activates the immune system so that if the plant is under attack from a pathogenic microorganism such as Pythium or Rhizoctonia or one of the root rots, the plant's natural immune system responds almost immediately. Why would a microorganism stimulate the immune system of the plant? To protect its food source. They're working together in a mutually beneficial way. Also, microorganisms make digestive enzymes right on the surface of the roots. Now, plants can't take up large organic molecules. Those large molecules have to be broken down into smaller and smaller particles until they're in their ionic form. That's the form that plants actually take them up through the roots. So microorganisms make the digestive enzymes to make the minerals more available to the plant. It's called mineralization. Protease, it breaks down proteins, some of which are just slough from the cells of the plant as the roots are growing through the soil. It breaks down the proteins into amino acids. Now, two of those amino acids, glutamic acid and glycine, are intermediate chelators. Chela means claw. So a chelate is an organic molecule that attaches to a mineral like a claw. It holds it tightly enough so that it doesn't lock up with other minerals in the soil, but loosely enough so it's available to the plant on demand. Microorganisms also make organic acids. As they feed on the sugars, they make what are called Krebs cycle intermediates. The Krebs cycle is cellular metabolism, respiration, to change the carbohydrates into energy. But in the process, they make organic acids, such as citric acid and malic acid. And those organic acids lower the pH in the root zone. The surface of the roots could be 2 points lower in pH than the surrounding soil, improving the uptake of the transition metals such as iron, copper, manganese, and zinc by the plant. Also, some microorganisms like Bacillus subtilis, GBO3, make a special chelator called a siderophore. Siderophore literally means iron carrier. It's one of the best chelators of iron in nature, so it keeps the iron soluble and available to the plant. And finally, some microorganisms make volatile organic compounds, a bouquet of aromas. So even if the microorganism doesn't touch the plant, doesn't even touch the root, it makes a bouquet of aromas that can stimulate up to 600 genes in the plant. One of those genes tells the plant, take up more iron. So not only do microorganisms make the iron soluble, make it available, they also tell the plant to take up more of it. As the plant takes up iron, it stimulates the plant to make more chlorophyll. makes more chlorophyll, harvests more light energy. Harvest more light energy, makes more sugars. Makes more sugars, some of them are used for energy to grow, to reproduce, to make more roots. Some of them are stored in the flowers, in the fruit, for the next generation. Some are leaked from the roots as exudates to feed the plant growth promoting rhizobacteria. So the cycle is complete. The plant feeds the microorganisms, and the microorganisms in turn help feed the plant. There are two main ways to add soil biology to the root zone, either microbial inoculants or compost teas. Now, the secret of a high-quality compost tea is high-quality compost. You want to have compost from a variety of sources. You want the compost from farmyard waste and also from worm castings. Now, just because there's a compost in your soilless mix or in your soil, it doesn't mean that the microbiology is rich in that compost. A lot of the composts that are made commercially are produced in large windrows. So the center of the compost gets very, very hot, and it will kill some of the mesophiles. Those are the microorganisms that grow at medium temperatures. Some of those mesophiles are some of the superstars of the rhizosphere, especially for stimulating the immune system of the plant. The actomycetes are one of those that are mesophiles. Also the trichodermas, very powerful microbes in the rhizosphere, one of the main superstars, but they grow at moderate temperatures. So even if you have a potting soil with compost in it, it will behoove you to add microbial inoculants to that soil. The compost tea will add diversity, wide diversity of microorganisms, and it also adds some fertilizer value. As the microorganisms are digesting the compost, not only are they making the minerals available, they're manufacturing some of the biostimulants that we just talked about. So compost tea goes way beyond just the benefits of the mineral nutrition alone. It's stimulating the uptake of those nutrients as well. Now, the downside of compost tea is that it has to be actively aerated. The aerobic microorganisms are the good guys. The anaerobic fungi are the bad guys. The anaerobic fungi grow in stagnant water without oxygen. Those are the root rots, the rhizoctonia, the pythium. They attack the roots, break them down. They're pathogenic to the crop. And that makes it so the plant can't take up minerals efficiently. You'll see symptoms of nutrient deficiencies in your plant on the top growth because the roots are unhealthy. But microorganisms protect the root cells and actually make antifungal and antibacterial agents. But it has to be done in actively aerated compost tea. So you want to make sure that it's continued to have a bubbler in there or that the water is circulating as you're brewing your compost teas. The other way to add soil microbiology is microbial inoculants. Now, the secret of a good microbial inoculant blend is choosing microorganisms that complement one another instead of compete with one another. In a compost tea, you're going to have wide diversity of microorganisms. Some will work together, some won't. But in a microbial inoculant, you can pick just the superstars of the rhizosphere, those with superior characteristics. I work with one microbiologist out east. He has 1,200 strains, pure strains of plant growth promoting rhizobacteria in the lab. And out of that 1,200 strains, he can combine the ones that are the best of the best at what they do. Some are superstars at taking nitrogen out of the air, changing it into fertilizer from the plant. Other specific strains are better at unlocking phosphorus. Others are better at making the root growth hormones or the other growth hormones for the plant to stimulate root growth. Others are a little bit better at making plant protection agents or rescuing the plant from stress. So if you have 1,200 strains harvested from the most fertile soils and from disease suppressive soils, you can pick those that just have the genetic superiority in their characteristics and then combine them in a way so it's a dream team to inoculate the rhizosphere. Another benefit of the microbial inoculants is that they have a guaranteed analysis. Now, it's not only saying what's there, but also what isn't there. They're grown in the lab under controlled conditions with quality control. In a compost tea, it can be very, very beneficial. And some of you are masters at making your own blends. It's a science and an art. But this is more of the science. If you have an improperly made compost, there could be pathogens in there. There could be E. coli. There could be salmonella that cause human diseases. Just because it's organic doesn't mean it's good. There's good guys and there's bad guys in the soil and also in compost. But in a microbial inoculant, you won't have any of those human pathogens or pathogens for the plants. If you look at the label, you can see what the guaranteed analysis is. Each of the microorganisms will be put on the label as CFUs. CFU stands for Colony Forming Unit. To be a CFU, of course, the microbes have to be alive, and they have to be able to form whole colonies. If you look at a petri dish, you see the discs that are on there, those are colonies. So that's how we test it. It has to be guaranteed amount. Another benefit of microbial inoculants is you can put together specific blends for specific purposes. You could put together a blend that's dominant in the mycorrhizal fungi, especially if you're going to be doing all organics. On the other hand, you could put together a bacterial-dominated blend, even blends for specific stages of the plant's growth. You could have one blend that just colonizes the roots early on to improve the uptake of phosphorus, improve the root strike and establishment of the plant. You could put together another blend for the bloom stage that would have more of the microorganisms that produce plant protection agents or rescue the plant from stress or aid with the finishing power of your plants. Or you could put together a blend that's specifically designed for disease protection to help the plant recover from powdery mildew or from other pathogens in the root zone, because some microbes are superstars at making plant protection agents. So let's take a minute to look at some of the key players in the microbial dream team. We'll start with the phosphorus solubilizing bacteria. Phosphorus gets locked up easily in the soil. We talked about that. It locks up with phosphates, it locks up with iron, makes nutrients unavailable to the plant. But some microorganisms produce organic acids and enzymes such as phytase and phosphatase that unlock phosphorus from the soil, unlock phosphorus from organic matter, feed it to the plant. Now, some phosphorus solubilizing bacteria are synergistic with the mycorrhizal fungi. Remember, the mycorrhizal fungi inoculate the roots, then as they grow in the rhizosphere, they exude organic acids and enzymes to unlock phosphorus. Now, the plant growth promoting rhizobacteria, the phosphorus solubilizers, hitch a ride on the mycorrhizal fungi. It takes them to places they could never have reached on their own in the soil area around the rhizosphere. On the other hand, they swim in the soil solution to places even the mycorrhizal fungi can't reach. They unlock phosphorus from the soil, feed it to the fungi. The fungi, in turn, feeds it to the plant in the roots. So they're synergistic. If you have a starter blend with mycorrhizal fungi, make sure that it includes phosphorus solubilizing bacteria. One of the best solubilizers of phosphorus in the rhizosphere is a bacteria called Bacillus megaterium. You definitely want that one on your team.
Okay, I'm just going to pause right here because I'm going to forget what to say otherwise. But you heard what he said about phosphorus solubilizing bacteria. Why do you put phosphorus in furrow in the spring? Because it's needed early on, right? That's where the majority of it's needed and taken up. So and we'll get to that in the slides here, but the one of the things in the Biomentum is, of course, phosphorus solubilizing bacteria, but again, the bacteria is different than the fungi, and as he said, the bacteria cannot move through the soil readily. The fungi can, so the fungi goes and moves all over the place, but the phosphorus solubilizing bacteria, like he said, hooks a ride, solubilizes stuff, and brings it back. So we have, of course, 25% fungal in the Biomentum.
It's a giant in the microbial world. It also forms chains, massive enzyme producers, and it's excellent at unlocking phosphorus. Next come the nitrogen-fixing bacteria. Some microorganisms are able to harvest nitrogen from the air and change it into fertilizer for the plant. Now, with nitrogen fertilizers, they're made the same way, but we have to take in the nitrogen from the air under high temperature and high pressure in order to make those fertilizers, and they're extremely soluble. For example, nitrate nitrogen, you can feed it to the plant. If there's a big rain, it'll wash the nitrogen away from the root zone, and it could go into the groundwater and into surface water. But if you use the nitrogen-fixing bacteria, they're fixing nitrogen, they're manufacturing fertilizer right on the surface of the roots. It's 95% efficient. So some of those microorganisms work in a team. Some fix the nitrogen from the atmosphere and change it into ammonium. Other microorganisms change ammonium into nitrite. Others change nitrite into nitrate, and then the plant takes up the nitrate nitrogen. So we're sequestering nitrogen in the root zone and manufacturing it real time, right when the plant needs it the most.
Okay, so he's talking about nitrogen fixation, and we'll look at that a little bit more, but this goes back to these guys again. And this is the basis for your farming right here. And as you remember, you get 90% of these and 10% of these at best, okay? What we're trying to do is we're trying to increase the number of these, the fungal. The red represents the nitrogen, the black, the carbon. So in order to increase these, they have to eat these. So they're about 5 to 1. So these guys, these fungal, have to eat 5 or 6 of these to keep on living, okay? And that's why, and remember this number, and I know you won't, but you need to have at least a 16 to 1 carbon to nitrogen ratio in your soil. The best way to find that out is a Haney test, and we will get to that in the slides. But when we increase these numbers, for example, when this guy eats 5 or 6 of these, of this one, he only needs 1 of these. So there's 5 or 6 of these nitrogens that are released for plant use. And that's basically what he's talking about there as well. So what happens in the root zone, and the reason we're trying to raise fungal numbers in the root zone specifically, is through the process of raising the good guys that bring the food into the plant, we're also releasing nitrogen for the plant use. And that doesn't even include the nitrogen-fixing bacteria that's in the Biomentum, okay? So you're gaining from it 2 ways. The more of these you can grow, the more nitrogen you're going to release for plant use. And that's a really big deal. And like I said, that does not even include the nitrogen fixers that are actually there in the soil. Okay?
How much of an uphill battle is it using GMOs and all the chemicals we use to get that?
We'll get to that. That's what we're going to talk about once we get done with this guy. Okay? I want to tie that all together and show you guys what's happening, why, and what we're doing to fix that problem for you, okay? And that's why I have these guys from Nebraska here too, to talk about how we can measure that, because it's all BS without numbers, so.
Another thing to remember is that microorganisms such as bacteria rarely die of old age. They're eaten by other microorganisms like protozoa. A protozoa eats a bacteria. It uses some of the nitrogen for its own use, for growth and metabolism, but then it spits out the excess. That excess nitrogen is fertilizer for the plant. So we're sequestering and recycling nitrogen directly in the root zone where it's needed the most. Some of the microbial blends coming out of the universities, they found that they could reduce the nitrogen fertilizer in half, by 50% or more, and increase yields at the same time. That's revolutionary for our planet, not just to protect the environment from nitrogen runoff, but to help the farmers to be more efficient, to restore the soil.
One other thing. So we always show this slide of the organic matter in your soil, right? And one of the things they've learned is that organic matter, for every 1% organic matter you have out there, you have 1,000 units of nitrogen. So the more that you can break down, and the most of that you can break down with good biological activities, 3 to 5% a year, okay? But in organic matter, 50% of your organic matter is carbon, right? Remember we talk about that all the time? 50% is carbon. The other 50% are dead microbes. What do the dead microbes have? If they're 90%, if they're 90% bacterial, there's nitrogen on all of them. And so that's where the 1,000, and you've got billions of the bacteria out there in your soil. But what's tied up in the organic matter and on every bacteria that's out there is 5 units of carbon and 1 unit of nitrogen. They just need to be eaten by the fungal so they can be released. So you've got all kinds of resources out there. That's what we're trying to, the point we're trying to bring across.
When you work with nature, when you work with microbial inoculants, the soil actually improves from year to year instead of becoming more and more depleted over time. Other microorganisms are superstars at making plant growth hormones, such as auxins and cytokinins. In fact, one microorganism feeds on exudates from the plant and changes it into IAA, indole acetic acid. That's the rooting hormone. Plants exude carbohydrates, remember? They feed the microorganisms. One of the carbohydrates they exude is called tryptophan. It's an amino acid. Well, some microorganisms literally snip off an ammonium ion, just one ion from tryptophan, changes it into indole acetic acid right on the surface of the roots, stimulating more lateral root growth and more root mass. Other microorganisms are better at making the cytokinins. Cytokinesis means cell division, so cytokinins are the hormones that stimulate cell division. So you combine the two in the dream team, those that make the auxins with those that fix the nitrogen, with those that make the cytokinins, put them together, it's synergistic. You'll get more lateral root growth, more root mass, better uptake of water and nutrients by the plant.
We're going to stop it again. So this is the analysis, so on the left-hand side, of the amino acids. And as you will notice, if you look at the amino acid profile, he was just talking about tryptophan and the importance of that in root growth. One of the amino acids, if you go down the center of the sheet, about almost all the way to the bottom, tryptophan is one of the things that's in there. The other thing I want to point out is he talks about glutamic acid and glycine. They're near the top of that same list, and those two amino acids are what bring calcium into the plant. They know that. And those two, glutamic acid and glycine, that's another reason why we use the amino acids, because of the wide variety of not only amino acids and what amino acids do, but it's a very good source of bringing calcium into the plant. Okay, I got more, but we'll continue on here.
Some of the microorganisms that colonize the root zone are also endophytes. They go into the plant. They penetrate the roots, go into the stems, into the leaves, into the buds, and process and produce biostimulants. They make phosphorus more available, they make nitrogen more available, and they even make growth hormones inside the plant. So it's not just the surface of the roots. It has an effect long-term on plant growth and health. Other plant growth promoting rhizobacteria are prolific enzyme producers. Remember, plants can't take up the large organic molecules, but microorganisms make digestive enzymes that break down the large molecules into a form the plant can take up. But they do more than that. If you find the right blend, you'll have those that feed each other while they're feeding the plant. For example, some microorganisms make the proteases that break down proteins into amino acids. That's a nitrogen source for the microbes. The other microorganisms are prolific at making cellulase. They break down the large molecules of cellulose into smaller sugar molecules. Combine the two, one's making nitrogen to feed the one that makes the sugars that feed the other. Those make sugars that feed the ones that unlock phosphorus. They provide phosphorus for the ones that fix nitrogen. You see, it's a team. It's not a single microorganism. It's a combination that feed each other while they're feeding the plant. Also, the byproducts of the digestive enzymes of the microorganisms have a direct effect on the plant. For example, the ones that make nitrogen available by producing amino acids aid with the uptake of calcium by the plant. Glutamic acid and glycine chelate the calcium and stimulate root cells to open calcium ion channels. The plant takes up more calcium. It strengthens the plant against fungal diseases on the leaf. So microorganisms in the roots make the plants more resistant to fungi like powdery mildew, bud rot, and other fungal attack on the leaves. So while microorganisms are feeding on carbohydrates, they make organic acids called Krebs cycle intermediates. They're byproducts of cellular respiration, cellular metabolism, and it has a direct effect on the plant. The Krebs cycle intermediates actually make some of the trace elements more available to the plant. They lower the pH in the root zone, sometimes by 2 or even 3 points, and solubilize minerals such as iron, copper, manganese, and zinc. Those trace elements activate enzymes that the microorganisms make and activate enzymes in the plant as well. If an enzyme is activated, even one molecule could do over 1,000 chemical reactions per second in the cell. Other select strains of plant growth promoting rhizobacteria, some of them are a little bit rare, but they're there in the environment, actually improve the photosynthesis process itself. The way they do that is by producing volatile organic compounds called VOCs. They're a bouquet of aromas that are very volatile that stimulate up to 600 different genes in the plant.
I just want to point out one thing. So when we do the soil tests in furrow and between the row, there's usually a definite difference between pH. And as he said, the pH makes a lot of difference on uptake of nutrients as well as trace minerals, okay? I actually have a little pH meter. It's got a probe on it about that long and it's got a dial on top. And we did some of that this summer. We'd go out there and we'd stick that thing between the row and then in the row and read the pH difference, and there was a definite difference, at least 1 point, usually up to 2, depending upon what the soil pH was originally. But that's what the biological activity does in furrow. It lowers the pH, and that's what you need because of this right here. And we've talked about this many times before, and that's the reason I have this here, is, you know, a lot of you guys lime or whatever, and you say, so what if my pH is too low? It doesn't matter if you've got good biological activity in furrow, because this is a sweet, it's right here, sweet spot's right here, and that catches most everything. And that's about a 6.5 pH, as you can see. Right here, about a 6.5 pH. And then if you go up that line, that catches, it's not in the middle of everything, but it catches about everything. And most of the time what we see in furrow when we check pHs, when we send it into the lab as well, that we're right in that 6 to 6.5 range mostly. And that's going to be where you get your best uptake. And so that's actually a very big deal when it comes to nutrient uptake.
One of those volatile organic compounds tells the plant, take up more iron. So they're making Krebs cycle intermediates that make the iron more soluble. They're making siderophores, which are very good chelators of iron, and then they're communicating with the plant. Even if they don't touch the plant, they're telling the plant, take up more iron. The plant takes up more iron, remember, makes more chlorophyll, harvests more light energy, makes more sugars, growth, reproduction, energy to fight off diseases, and also extra carbohydrates to leak from the roots to feed the microorganisms.
Iron is the central molecule for photosynthesis. It doesn't happen without iron. We did a lot of tissue testing this past year to check not only on iron uptake, but some of the other trace minerals. And the longer we got into the growing season, what we found out was the iron kept coming up. And hopefully that was because of biological activity in the soil. And so it's very important to have good iron intake. Most of the time there's all kinds of iron in your soil, but it's locked up with phosphorus, just like he was talking about earlier. And the plants need iron. It's vital for photosynthesis.
Another one of those volatile compounds also turn off the sugar signal inside the plant. That's really great for the finishing power of plants. When plants have enough stored carbohydrates to make it to the end of their life, they stop doing photosynthesis. They stop leaking carbohydrates and the microorganisms start to go dormant, go to sleep on the root zone. But microorganisms want to live, they want to reproduce, they want to have more colonies formed. So they make a volatile compound, shuts off the sugar signal so the plant doesn't know it has enough stored carbohydrates. So the plant will continue to do photosynthesis, it'll continue to bulk up and sweeten up and grow all the way to the day of harvest. Another benefit of the increased photosynthesis is even at the early stages of growth. If the plant makes more carbohydrates than it needs to use for energy to grow, it sends some of those extra carbohydrates to the roots. It's stored as starches in the root zone. Some of those starches are used for energy by the plant for active transport, to take up elements such as phosphorus and potassium. So if you have more stored carbohydrates in the roots, the plant will take up phosphorus faster. Phosphorus stimulates earlier flowering, more flowering sites. If there's more stored carbohydrates, you'll have a better uptake of potassium for carbohydrate metabolism to unlock the stored sugars so they can be shipped to the flowers and be stored in the fruits. So if during the vegetative growth stage you enhance the photosynthetic capacity, you'll have a faster transition from grow to bloom. In the lab, I've seen it. With high brix plants, with lots of stored energy, the transition was much more fast, much steeper than plants that were in the control group. In fact, in that early transition phase with the high brix plants, I had 20% more flowers in the early flowering stage. Finally, there are other select microorganisms, superstars of the rhizosphere, that are excellent at providing stress protection for the plant. They rescue the plants from stress. When a plant's under stress, heat stress or salt stress, drought stress, the plant produces ethylene. And ethylene downregulates the root growth and the shoot growth by the plant. But some microorganisms are ACC positive. They make an enzyme that breaks down the precursor molecule to ethylene. So it rescues the plant from stress and restores normal root and shoot growth.
Okay, we covered a lot of stuff there, and a lot of the stuff he talked about in there is what we're going to cover the rest of the day here. So as soon as we get the PowerPoint back up, we will continue on. Okay. Again, this is a reminder of what happens in your soil with phosphorus tie-ups. And I know everybody puts phosphorus on every year because they're always short on it, right? Are you short? So between 6.3 and 6.8, that's the ideal pH range. But like we were talking on the board here, but low, if you have a low pH, your phosphorus is going to tie up with aluminum and iron. Okay? Have any of you tested your soils for aluminum? Hardly anybody does. The standard Midwest Labs test does not do it. You have to ask for it. It's not, I think it's like 30 or 40 bucks. I don't remember right off. The guys that are going to talk to, you guys test for aluminum, Austin?
No, it's not standard.
Not standard? Okay. We'll talk a little bit more about that. What contributes to high pH, however? How many of you have high pH on your soil? Okay, a number of you do. This can be contributing factors here. Poor drainage, compaction, and reduced biological activity. So if you notice on a soil test, there's sometimes the Olsen and sometimes the Bray. So for those of you that don't understand what that means, the test used to determine phosphorus content above the pH of 7 is the Olsen extraction method. Below 7, it's called the Bray extraction method, and you'll see that on your soil test sometimes with different soils. So P1, of course, how many of you are familiar with P1 on a soil sample? Y'all, I'm sure you all are. P1 is what's plant available. P2 is what's out there, but not been converted into what would we call plant available. Just want to review that and make sure you understand that. Not that that's going to make a lot of difference going forward here with what we're looking at. So this is what you're dealing with in your soil. And if you have a high CEC soil, that means a lot of clay. Low CE soil, of course, means more sand. You're dealing with all these positively charged ions around this soil colloid. Why? Because the soil colloid is what charge? It's a negative. All that, and so CEC, of course, is a determination of how much clay particles you have out there in your soil, right? That's what your CEC is. So the higher CEC you have, that's a good and a bad thing. The good thing is you can hold way more nutrients in solution in your soil than with a lower CEC. The bad part is you got to be able to pull that off the soil colloids so that it's plant available, because the plant cannot do that by itself. So oops, went too far there. Where did we go here? Okay, so this is what you're dealing with in your soils, depending upon your CEC. Now, we're going to get into, and I'd like you to turn to this first slide, please. Dig this one out. And the reason is, this is a soil sample from a producer up on the Minnesota-North Dakota border. And this is what his numbers looked like, and I wish I knew, and if I could ask Ron Kaufman, he could tell me, but what we want to look at here is the P1s, and it's one of these numbers, and I'm not sure which one it is, that we're going to be looking at more in depth, but we're going to just look at phosphorus, because phosphorus is what he was talking about, and phosphorus is a biggie. So let's go to the next slide. Okay, beet lime. So this producer could buy could get all beet lime free of charge, high in calcium. All he had to do was haul it 50 miles away. And we told him not to do that because of the top column right there, aluminum. Aluminum, 1,219 parts. Okay? Very, very high. Now, calcium holds on to, you heard what he said, calcium and phosphorus join up to make calcium phosphate, right? It's the same thing your bones are made out of. It's insoluble. So that 90% of the phosphorus you're putting out there every year, that's what happens to it. It ties up with that, or aluminum, or with iron. Okay? In this case, these guys put beet lime out there, even though we told them not to do it. And let me tell you, the first year, the available calcium in there, because see, the calcium in there was 21%. And that's what they were after. So their calcium, the first year, they had tremendous results. But the second year, this is what happened. And if you go to the next slide and you look at the P1, their phosphorus was 8. It went down to 8. And it was at like, it was like 60 some, I think, on the originally. But it went down to 8. You see where the P1 is on there? Okay, what happened was the aluminum tied that up. And so they had no available phosphorus and their crops went to crap. So we said you have two choices. You can either you can either go buy a tanker load of acid, like hydrochloric acid, and go out there and spread that on to break that bond, or you can go out there with a pure calcium. So they opted for a pure calcium, and pure calcium they put on was Pell lime. So if you go to the next slide, the spring after 1,000 pounds of Verlyn. You see the, where'd we go? You see the P1 on that? 103. Now that didn't come from the calcium. It was there all the time. What the calcium does is the calcium displaced the aluminum on the soil colloid particles. Remember, we looked at the colloid particles. That's why I put that up there first. There's aluminum on there too. It doesn't show anything on there, but there's aluminum on there too. And aluminum, charge-wise, is 10 times stronger than calcium, because aluminum is a +3, calcium is a +2. So we had to kick the aluminum off there and displace that so that the phosphorus could be freed up in the soil. Very simple.
Where does the aluminum go?
The aluminum stayed in solution in the soil.
Tied to nothing.
No. It tied up with something else. It had to tie up with something else. Either that or was in solution, but they did not put on any more beet lime, I can tell you that.
So when this here Verlyn starts reducing through the years, the aluminum starts tying up again?
It will, yes. And those number, that P1 number will start to go back down. Once that is there, you do not get rid of it. Aluminum does not go down through the soil surface because of its charge, strong charge. It stays there. So once you got the problem, you got to deal with it every year. That's why these heavy metals are the same way. And we discourage guys from putting this sludge on from some of these municipalities because of the heavy metals in there. It's cheap, but you're going to pay for it the rest of your farming career. That's the bad part.
What about that acid you were talking about? That
It would release the bonds, but the aluminum's still there. Yeah, that's not a good option, actually.
What about putting Verlyn on high pH? What's that mean?
So, what you will do, depending on, that's kind of a loaded question, because there's a number of things that could happen, okay? In some cases, you could lower the pH. In some cases, you might raise it. We have done in the past, it's been years ago, we have actually treated high potassium soils. You can have a very high potassium soil, just like all of you have very high iron in your soil. And what we did was we treated like with like. So what we did was we put potassium on. It freed up the potassium for the soil to make it more available. And because calcium is so important, you might get the same response depending upon what your magnesium and phosphorus are. So I didn't answer your question.
Well, that's fine. I had a neighbor, they did that.
Yeah.
And they said it just woke up the soil and real. I go,
Yeah.
really? I think you could.
It woke it up. It does, because it can go one of two ways, depending upon what else is there, yes.
Flip the coin?
You got to kind of know what you're dealing with, but no, it's not necessarily flipping a coin.
Okay.
There's other things involved. When you start talking chemistry,
Okay.
when you start talking about biological activity, that's a whole different story. Okay, so this is the chemical side of what happens in your soil and what can happen. It's just the chemical side with mineral tie-ups. That's only one part of what we're going to discuss here. Okay, so I'm not sure if you guys have seen this before, but this is a cornfield down in Georgia, and I've talked about this before, but I'm not sure I showed you a picture before.
Yeah, you did.
Did I? Okay. This is a cornfield down in Georgia. Amaranth pigweed down there is unstoppable. They can't, Roundup doesn't touch it. Okay? So this looks bad, I know. This is grass in here, but if you look right here, you look right there at the bottom, those are amaranth pigweeds. And what we did with this field is we spread calcium on that. And the reason I'm bringing this up is because if you look at this book, When Weeds Talk, in there you can look up any weed you want, and the first column in that book is calcium. And it has a mark by each one, and for amaranth pigweeds, calcium is low, just like for 95% of the weeds out in your field. Okay? I'll pass this around. You can look up your favorite weed and you'll see. So what we did here is, because we knew this is the other side of the field. And you see there's a corn there, there's one there, there's one there. Okay? It was all planted at the same time. This is a cornfield. So what we did on this side of the field, we spread Pelline, calcium, because that soil was so deficient in available calcium. Woke the soil up. Amaranth pigweeds are there, for the first reason the amaranth pigweeds are there is because of the lack of calcium, available calcium in the soil. You all have lots of calcium in your soil, but it's not available. So I want you to keep this in mind. We'll keep moving here. But this is right next to it. Right next to it. That's the difference. There's no herbicide involved on either sides of those at that point. And this guy is not worried about the grass because he can kill that easy. Okay, this is in Ohio. And this is interesting because this guy has been using the Biomentum for, 5-6 years now. He does not use any pre's on his corn or soybeans. He has changed the structure of his soil enough that he doesn't have to do that. But if you look up here, in every one of his fields, he had, what did he say? He had radar and GPS. Well, he didn't turn his radar on, and the GPS didn't start it quick enough, but there was fertilizer in furrow-biology, basically ABC, in those rows there. That's where the planter rows, not sprayer rows. And he said it showed up all season long. And this is another picture in another field. But there was no seed in those areas. So the reason I bring that up, and the reason he sent those to me and called me on it, is he said there's got to be a symbiotic relationship here between the plant, the crop, the corn that he was trying to grow, and the biology in the soil. And he had that, those fields were a weed problem all year long. But there was no pre on any of these fields, and that's soybean field. And that's about 6 weeks after planting. Oops. That's about 6 weeks after planting. His biologically, he's changed his soil enough that his agronomist cannot believe that he can get by on the herbicides that he uses, because he doesn't have to use much anymore. So what's happening is he's changing the biological balance in his soil.
Is he applying any calcium?
No. He's not. And they have very high make soils out there. Yeah, when they tile out there, they get boulders. It's just unbelievable. Okay, how many of you use an insecticide? Okay. So we've got some slides coming up, but so this is a guy that uses index insecticide. Which ones do you guys use, by the way? Bifenthrin. Okay. Rick? Okay. All right. Well, so I got 3 guys back down in northwest Iowa. They all put insecticide on. And one does corn on corn, the other 2 do not. All 3 of them got burned about 3 years ago and had corn laying over. And so they don't want that again. It's no fun combining if you've ever done that. And so they use an insecticide. This guy is using Index, and it costs them $24 an acre. So what we did and what we're doing, this guy is corn on corn silage every year. And so what we did is we took an in-furrow analysis of the biological activity, because we wanted to see what was going on. And if you see up here, the fungal was at 6% and the bacterial was at 94% in furrow. That's what the IF stands for. Normal is 90-10, right? Okay, we took the same thing between the row and it was 12% and 88. The fungal actually went up between the row. So what is the insecticide doing to the fungal activity? And this guy put a quart of Biomentum in furrow. He used the ABC. Next one, capture insecticide. This guy rotates. Okay? This was his, this was between the row, BV, BTR. This was corn, of course. And the fungal was 18% and the bacterial 82%. Actually, in furrow here, his fungal was 27 and 73. So the other thing that we're looking at is if you look at this number up here, the UGC, that is the total bacterial activity in furrow. That's at 400. And if we go back, where's that one? No, that's the wrong one. Here, if we go back, if you look at the total number between the row up here, it's 174. You see that? So the total activity is down as well. So this is the right one. Yeah. So between the rows, his total activity was 225, 18 fungal, 82 bacterial. And if we go to the next one, 400 in furrow, 27% and 73. So that's actually, even though he's using a fungicide here, or insecticide, I mean, in furrow, he is getting some loss, but his numbers are way higher. The first guy is using a double rate of insecticide, and the first guy, by the way, last year we did on his corn on corn, where he has an issue, we put 2 quarts of the ABC in furrow on half the field, insecticide on the other, and he had no issues. You couldn't tell the difference. Where the line was, you couldn't tell the difference when we went in the field. So we're getting some biological activity, and I think, Clint, you've been with the program that you're on, you saw some possibilities this year as well with reduced rootworm numbers.
Yeah, when we show the program,
You'll show that? Okay.
well, I don't know if he'll show my field specifically, but we test for the corn rootworm eggs in the soil. And
Good.
fields that I tested last year were high, and on corn this year, those numbers are back lower. But being able to use this program, we're able to see that. Now, I don't know if it's all just because of this, but it just seems like it was pretty cool to see. This field's been corn on corn since I'm 42 years old. It's been soybeans maybe 2 or 3 years of my lifetime.
Oh,
It's not very friendly for soybeans because it rocks, so we've always just planted corn on it. But it's pretty cool to see the differences that we are changing from year to year by using the Biomentum. So, yeah, I feel like it does help with the rootworm. And I don't know if it's, like, I don't have rootworm issues. I haven't had a case where I've seen rootworm like devastate a crop. Now, I've been using biologicals quite a while, not just the Biomentum, I've used mycorrhiza as well, and my root masses are getting bigger. So you get a bigger root mass, it takes more stuff to affect that root mass. So if you keep even if you have rootworm out there, you might not see the effects of that either. So, but I feel that it does help quite a bit on the rootworm. So.
well, and we'll find that out because we want to do this ongoing year to year, but in Nebraska, rootworms in areas, I understand it's a big problem.
Northern especially, even guys that rotate. I actually have a customer back at home. It was beans last year, and we sampled it here this fall, and surprisingly enough, 180 of the quarter has really high rootworm pressure. He didn't realize that, and I know 2 seasons ago in that same area, there was a lot of guys, the Pioneer Seed guys, that were having a lot of rootworm issues, and they're finding out it's because of Northern and the extended diapause. So I don't know if you guys have Northern up here, but the Northern, they're finding out can winter 2 years now. So even if you rotate, you might still have that problem. Well, he didn't know it was there until we sampled. So now he can either put an insecticide on or bump that half of that field up to a Smart Stacks or something to kind of help combat it. So it's very eye-opening what we're seeing with it and what we're learning. So like, he'll explain it more, but yeah, everything's just getting to be more and more of a pain in the butt. And if you can test for it and try to figure out if there's actually some of my fields, I ordered my seed before I did my soil sampling after harvest because it's a better price. But now I go back and I do the rootworm testing on a lot of my fields. I don't have high pressure, so now I'm kind of wishing I would have bought the double pros. And I guess, I don't know, they don't like to downgrade their seed once you purchase the Smart Stacks, because I'm all corn on corn this year, so I wanted to make sure I had plenty of protection. So yeah, but I think moving on from year to year, I think I'm going to be more mindful of that, moving forward. So
You guys are gonna like what these guys have as far as what their program shows. We'll get into that after lunch a little bit more. So I just want to point out that it's not just the fungal we're looking at, but if you take 27% of 400 versus 9% of 127 like the other guy had, that makes a lot of difference. So we're probably getting close to lunch here too. So this is another guy. He uses Aztec, and he's running it at half rate, which is $16. Kind of runs it in furrow, as it's closing. It's a dry product, of course. And this was between the row, and this was the total number between the row, 13 and 87. And this was in furrow. And again, he had around 380 total units here, but it was pushing 40 and 61. So this particular insecticide, he's not had any more rootworm issues, but this one in furrow, so to speak. We're getting the fungal numbers up. We're not at 50-50, but that was pretty darn good. The reason we're looking at this is, we're using all these different chemicals, and we're using all these different chemicals, and so we're putting pressure on all the biological activity in the soil that you need working for you. And so whether that's through tie-ups, like we were just looking at, or whether it's through an insecticide or a fungicide or a herbicide, there's all these different issues that you're having to deal with. And the sicker your soil gets, the more of these things you're having to use. And they all cost money. Okay? So this is another one that I wanted to, what did I want to look at it here? Okay, if you pull this one out, please. Are they ready? Okay. If you pull this one out, you notice on the left there's in furrow and between the row. What we're looking at here basically is we're looking at soil pHs. And this guy has been using the Biomentum for 3 years. Last couple years in furrow, and then he's been coming over the top for 6 years, I suppose. But his soil pHs are right in here in this range where we want to see them. And a lot of the numbers on here are very close as well, which is not normal either. So if you look down here on the bottom, sulfur is one of those that is a negative charge, and it doesn't move as fast through the soil as what nitrogen is, but it's just as mobile. And if it ain't there, you're not going to get it. But it's interesting. He had a higher level in furrow than between the row on these soybeans, but it was the same on the corn. And the reason it takes so much sulfur on soybeans is because sulfur is involved with making nitrogen available to the plant. You have to have it there. Let's see. Yeah, okay. So, this guy was talking earlier on this film about culturing all these bacteria and stuff, but fact of the matter is less than 1% of the microbes in the soil are actually culturable that you can grow. So this is what he was trying to emphasize, that the plant needs the microbial activity in order to get the nutrients. And so this is what we're trying to bring to you today on how to help you increase your profits. And I mentioned this earlier, the majority of microbes in the soil are in a dormant position. So this is why we use these three together: available carbon, amino acids, and the biology. We looked at this. That's the label. This is an interesting slide if you pull that out, and then we'll have lunch after this. This was a guy in Nebraska. This was he used a company called Pattern Ag, and that's what these guys' company used to be called. It's now called Earth Optics, right? So what he did this year, first time using the Biomentum, he sprayed twice, 8 ounces on June 14th over top the corn, and at V7, 32 ounces. And what he found out when he sampled his corn was this fall, his microhygia fungi was 27% higher, which he was pretty excited about. Phosphorus solubilizing bacteria was 25% higher. Anaerobic activity was 57% higher. You remember what this guy said on that film about anaerobic activity and how detrimental that is to nutrient uptake? So this is a big deal. Denitrification was 7% less. And I'm not sure what this means, but Austin may be able to explain something like that, but the guy thought that was a pretty big deal. There were no plant growth promoters that were noted to any degree. So, all right, we'll stop there, and after lunch, we'll have these guys give their presentation, and so you can see what kind of a management tool that can be for you. And then we'll finish up with some water stuff, and then some more of the material. Okay, we better keep moving here. Okay, we already mentioned this. We'll get back to the slides here, and then we'll finish up. We're talking earlier, and I know you guys have probably seen this before, but we're talking earlier about efficiency and what you have out there in the soil. So if you would pull this slide out, please. This is a trial we did again, where a guy sprayed 8 ounces of the Biomentum at V2 or 3 on his corn, and he did a sprayer with and a sprayer without. And so what he did here, if you look there on corn 1 and 2 on top, those are V6, and you can see no momentum and the momentum up there. The date tested, and then nitrogen, it shows 3.95, 3.98. If you go across there, there's not much difference on any of these nutrients at that point. Now if you go to V9, which is what? How much difference between V6 and V9 time-wise? 3 weeks? 2 weeks? Depends on conditions probably, but it's growing pretty fast then. So at V9, if you look at it again, the nitrogen on the no momentum was at 3.75, and with the Biomentum it was 4.82, and you can go across the same way. Potassium 3.8, 2.61, 0.25, and 0.35 on sulfur. Where'd that sulfur come from, and where'd that potassium come from? My point is it was already there. We just need to get it into the plant. And of course, these were tissue tests. But if you look across, magnesium 0.21, 0.26, 0.35, and 0.5 on calcium. Remember we talking about how important calcium is and getting that into the plant? So there you have it. And all of those nutrients, it's the same way. There's a difference in all of them. They've all gone up, okay? So the second part of that was at that time, when we start talking about nitrogen, and that guy on that film was talking about nitrogen, and we were talking about nitrogen as well, and how the nitrogen is released, but also what happens biologically. So what happened here, and this is a big deal, the organic N, which is a stable N, without the, where am I at here? Was it 440, no, here, that was soybeans. The top one, 41.73 and 58.34. 58.34 was the Biomentum, and then the inorganic was at 202, which is in the form that it can go up or down, and 117 with the Biomentum. And so the as Austin was talking, here's the WEOC scores on nitrogen, and what do you think about that, Austin? Is that good or bad? The Biomentum was at 50 and without was at 28. Which one would you rather have?
Oh, probably the 50, but the ratio I'd like to see a little bit better than we are.
Explain that, please.
Kind of carbon and nitrogen. Looks like the WEOC's the same, but your nitrogen's higher, so that's a little bit out of balance. Kind of want like a 16-to-1, but it doesn't. I don't mind those scores, but yeah,
Just be honest, come on.
yeah, I'd want to see a higher WEOC score, or a lower WEOC. Just so it's more balanced.
On the nitrogen or the
Yeah.
carbon?
Either or. I kind of
Okay.
like to have that ratio be a little bit different. About 16-to-1.
Yeah.
So.
Oh yes, the C-to-N ratio here is 5.2 versus 7.09. That's what you're talking about?
Yeah, I'm looking at
Yeah.
the CN ratio.
Yeah.
So.
Yep. Okay.
So the iron one count, is that good or bad?
On here,
Yeah.
this is the soil sample now, not the leaf sample. If we go back, is there iron on here? Yeah, there is. Whoops, come on. One more, okay. Here, if we go to the iron here, 281 in the leaf sample versus 206. So the iron is actually higher in the leaf sample, which is, again, that's a big deal because that's what fires photosynthesis and sugar production.
What rate was that put on, Verlyn?
It was put on broadcast with his post herbicide at 8 ounces per acre. That's all, 8 ounces.
10 gallon total, 10 gallon mix,
Pardon me?
10 gallon per acre mix,
10 gallons per 8 ounces per acre.
with a 10 gallon.
I don't, with what?
How many gallon carrier?
I don't know.
Total?
I don't know what his rate was for his post herbicide. I'm not sure.
I suppose V6 was with herbicide and V9 was without it.
No, V6, that was just a leaf analysis. That did not, that was just a check to see how many of the nutrients had gone into the plant at V6. Okay? And basically there was a wash, if you want to, there wasn't much difference there. It took long enough for that biology to translocate down in the root system and start getting things working. And so by V6, that wasn't working yet very much.
Oh, so there was only one application?
One
Oh,
application.
I thought there was two. Okay.
No, one application
So when did you put that on?
at V2, V3 with his post
That
herbicide.
long to show,
Oh, yeah. Yeah. And when you stop to think, 8 ounces broadcast, there wasn't much got into the plant. But that was the result.
Is that your recommended rate, or is that just what this particular one was using?
You know, the reason he put 8 ounces on is because the 5-6 years before that, when we did applications in the fall during fungicide time, you know, where we'd use fungicide and Biomentum and then the two together, it was always at 8 ounces. That's the rate we always used, okay? So that's what he used here too, was just 8 ounces. I got guys, they use, you know, we tell guys every time you go across the field, you know, 4, 6, 8 ounces, you know, because it's so dang cheap. And this is what's going to happen if you do.
Okay, well, is there a problem if you, let's say, double that, put on 60 ounces, are you gonna cause problems?
No,
Okay.
no, what you'll do is you'll stimulate everything in the soil. And with the 8 ounces, with the increase in organic nitrogen, this guy was an agronomist for 15 years, and he's scratching his head how that could happen. but you know how he was trained.
Yeah.
And so, but the organic nitrogen is very important because that's stable, of course. But normally, you know, we say 4 to 8 ounces if you're going to go with a herbicide, fungicide, whatever. I have guys that tell me, no, you should be putting on 16 ounces, not 8 ounces. You're going to do yourself way more good because of what it's going to do in the soil. I don't know if you're going to have any more activity in the root zone because we haven't done any side-by-sides that way. But you're just going to help everything in the soil become available as well. So, okay, let's see. Okay, so let's go to this slide. So all of you, if you haven't, probably have all used Roundup. And so we're just going to let all the skeletons out of the bag here and talk about what's going on in your soil and why. That's what this is about today. Okay, so this is foliar application of glyphosate up here, systemic movement through the plant, chelation of micronutrients intensifies stress. This is what happens. You take care of the weeds, but this is what happens, and you'll see why I'm telling you this. So accumulation of glyphosate in the soil, it's slow to little degradation, and you all know it takes 22 years for half of it to break down in the soil. So you've got residual in the soil and residual effects. The glyphosate is toxic to nitrogen-fixing microbes, which we are trying to build up in the soil. That's been proven. Bacterial shikimate pathway it shuts that down, and you've all seen the picture of 2 pots, one with a sterile soil with a plant growing in it and one with regular soil, and you spray them both plants with Roundup, and the one with the biological activity in it, but not with the sterile soil, will die because it shuts the immune system off. The one with the sterile soil, because there's no bad organisms in there to kill the plant, it stays alive. Okay, so that's what that is talking about, bacterial shikimate pathway. It's toxic to microhygia, which we're trying to build up, and biological control organisms, earthworms, and PGR organisms, which stimulate the plant, growth organisms. So it accumulates in the roots and shoots and translocates from the root and shoots into the rhizosphere. So what happens is you have what's called AMPA, that's the toxic part of it, but what happens is you have an increase of soil-borne pathogens, many of which Austin was talking about. Okay? That's what happens with glyphosate. Why have we seen the increase of all these soil-borne pathogens in the last 10, 15 years? Right, Austin?
Yeah.
That's what's happening. All right? I'm just trying to explain to you part of what's going on in your soil out there, and it's not coming from just one source of what's going on out there. So it also is hard on nutrient oxidizers like iron, manganese, and nitrogen. Well, we know how important iron is. That's how you get photosynthesis. Microbial nutrient sinks like potassium and magnesium. You can't get those into your plant. You can't get them solubilized and into the tissue because it kills off that bacteria that processes those. So it reduces the availability of these nutrients right here. And I'm not picking on Roundup. That's not it. I'm just trying to give you the whole picture here on why you get so frustrated on why you can't get things to work. So this is just percent decrease in mineral nutrients in corn silage from 2000 to 2010, about the time that Roundup started being used, and you can look at that in your leisure. This is quite interesting, and I threw that in there for you for reference. This stuff was all done before Monsanto put a clamp on everybody and said, you have to sign this document that you're not going to do any testing or we get your farm. So you can no longer get a hold of this, or you can no longer test, or they get your farm. That's what it comes down to. But this, for example, is Roundup Ready hybrids and different chemical herbicides here, and the different numbers of corn, and the difference in yield. And this is using glyphosated V6 and these others that did not use glyphosate, but these other chemicals on here, and a difference in yield. So I just thought I'd throw that in there for you, just as a reference. But this is the good side of it. We know this because this test was done in manure, and you guys I've made reference to this before, but as you can see, where we put the Biomentum in the pit for 6 months, instead of 134 parts per billion of glyphosate, we ended up with 67. So we reduced that by 50% in 6 months. So we can break this stuff down, okay? Then, I was going to do this after this, but because I wanted to lead into that, but this little thing about herbicides, do they have a charge? You remember we started talking in the beginning about that colloid that had all those charges on it, negative and the negative colloid and all these minerals? Well, your herbicide's got a charge too, okay? And so I'd like you to take this home and take a look at it, but this is what they're talking about. Atrazine may have a positive charge in acidic soils, which increases its attraction to negatively charged colloids. Now you know you have to put on different amounts of chemical, especially atrazine, according to soil type, right? Everybody knows that because you've used it. I'm pretty sure anyway. So anyway, you guys can read this and use it however you want, but this talks about the pH and the different effectiveness of chemicals, and they just threw this little formula in there that nobody's going to do anyway to figure it out. And that's when I was going to do this little deal. But okay, just to recap here, this is all the different stuff and what it does. And the one thing that when you go down here to bionematicide fungi, it is one of the properties. It infects eggs, juveniles, and adults. That's why we're interested in doing the testing with Austin to actually prove that with corn rootworm. And it destroys pathogenic fungi, cell wall borers, and of course we've talked about these things up here. So, all right. Just a quick note on sulfur. How many of you use sulfur? Okay. How do you determine how much you need? Or do you just put it on every year?
Shoot from the hip.
Yeah.
Soil test and add a little more.
Huh?
Go by the soil test and add a little more.
Yeah. There's all different ways to do it, but I just wanted to throw this in here. So, should you spend money on sulfur? So there's lots of different opinions on this, and sulfur, what they say is has the greatest response in soils with less than 2% organic matter. How many of you have tried putting sulfur on to increase your bean yields? Okay, has it worked? There's a reason it will and there's a reason it won't, okay? And that's what they're talking about here. Of course, it's mobile in the soil, but depending upon how deep you test is gonna tell you whether or not you have enough in your soil or not. But this is an interesting little read because some years they have had response and some years they haven't. And that's why I wanted to throw this in here for you. Sulfur is very mobile for the most part, but sulfur is very important. So take a look at that and read through it. If you're thinking about putting sulfur on, know what it says, because the conditions vary as far as how effective it is. As it says, low organic matter conditions favorable for sulfur deficiency. Low organic matter soils, coarse textured sandy soils, soils with high water tables, or though there have been saturated fields would be the first target. And then timing is always important. You put it on too late and it's not going to do you any good, of course, either. So this is a little deal from Midwest Labs for those of you guys interested in trying to balance your soil by putting phosphorus, potassium, and all that stuff on. So I just included that. I'm not sure I agree with any of those ratios, but there it is. You cannot find that on their website anymore. This goes back, as you will see, it goes back to, where is it? Oh, it must got cut off. This is 2016, I think, and I haven't seen it since. So this is just a little deal that we were looking at for tissue testing. How many of you tissue test? No? Okay. Okay. Just a suggestion, and this is a suggestion why. So this was a little plot that I had by my warehouse. Planted corn on May 18. May 20, applied 140 units of N and 31 units of sulfur dry. June 4, foliated a quart of Biomentum, zinc, and boron, and 1.2 gallons of amino acids. Tissue tested on July 1. Applied another quart each of amino N, Biomentum, and boron on July 4. On July 30, another tissue test. Drone applied a quart each of Biomentum and boron. And this is what the tissue test looked like. This is the first one that we did. Nitrogen was sufficient, and this was July 1st. Phosphorus was way above. The top yield, here's a note for you. The top yield guys say that you need, on a tissue test, you need at least 0.4 phosphorus in order to get top yields. This 0.4 or above. This came out, and you see this is 0.42 here is what they consider normal. This came out at 0.67. There was no P or K on there. Now, granted, this was elderberries for 8 years or whatever, and the fertility levels were very high on this field before that. But potassium was high, magnesium was way short. This was on July 1. Calcium was way short. This was July 1. There was no calcium put on there. Okay. Sulfur was about right. Iron was low. It was at 142. Manganese was way low, and it was way low all year. Boron, we had put boron on, so that was about right. Copper was about right. Zinc was way above. And if you go to the next one, that was July 30, and you look, nitrogen was still high, phosphorus was still good, it was at 0.42. You go over to calcium, we were way low. This was 30 days later, the calcium was at 0.66. It was above. okay? Iron was way above as well. It was at 163 at that point. So I just wanted to, this is the reason we tissue test, to follow some of those. The nitrogen was still high, phosphorus was still good, calcium was still at 80, iron was at 215, and the corn was doing very well. The boron was at 16, of course. So to me, I know, Austin, that you probably won't agree with me that, you know, this isn't the best barometer, but I wanted to find out what was going on in the field biologically, because we had put ABC in furrow at a quart of each. Biologically, if we were getting the nutrient uptake. Okay? Was that good or bad, Austin?
No, it's good. I like plant tissue samples, just.
I know.
It doesn't give you a future outlook, but it does show you what happened.
Yeah, it shows you what happened. Yep. What's out there and what's happening, right? So, I think it's important, if you guys want to be efficient, you know, it goes back to this, to that one right there. When you're short on one nutrient and you're shooting for 200 bushel corn, and it's only because of one nutrient you're only going to get to 150, you just peat away what you put out there. Okay. So, so this, we're getting to the end here. Hang on. So this was Wallace's. This was Kaufman's, and this was in row with starter. The total biological count, and you see where we're at. Fungal 4654. That's darn good.
Is that with Biomentum? Biology?
No, that was between the row. I don't know if there was biology spread on there.
That says in the row.
This was in the row, but this one here was actually between the row. Okay?
Okay.
All right. But so in this set of rows, what we're looking at here is what happens with, what may have happened with starter fertilizer. Because this was 5 rows, between the rows, where we had put starter fertilizer in row.
I know biology.
Yes, and biology.
And biology,
Okay? So this was the same, 5 rows, only this was in furrow. And you see what happened to the total count and the fungal and the biology versus between the row? We went backwards.
That ain't supposed to happen.
No, it's not. But did you hear what he said on that video? When you put phosphorus in furrow, what happens to the fungal? The root sets up a defense, and it does not allow the fungal to attach to the plant. It's like it acts like a predator. Okay? That's why they suggest, if at all possible, put it alongside the row. Okay? This was another one. This was in furrow on this piece of ground. 242 total, 25 and 75. This was soybeans. This had Biomentum in furrow. This was between the row. Whoops, where'd that go? Whoops. All right. Okay. This was between the row. Nope, wrong one yet. Here we go. Between the row, and it was 373, 40, and 60. As a, oh darn it. So this is what can happen if you've got, if you're putting stuff in furrow biologically. Okay. Now, to give you another idea here, this was, the other ones were 8 ounces. This was 8 ounces, or I mean a quart. Where's this one? Here. Oh, okay. I'm going too fast here. All right. So this was in furrow. This had the Biomentum and the ABC in furrow on beans. This was a field that we were checking for biological activity. Between the row, these are the numbers. So what we're trying to determine is the biological activity continuing? So out in that same field, we found where the old corn row was from the year before. We could see little stumps, and we took samples to find out what was happening biologically in there, if it was continuing to work in the soil. You know, you put biology out there, is it dying or is it doing you some good? So this was the corn row from the year before, and this was the total biological activity out there, and the fungal was at 40%, and the bacterial was at 60% in the old corn row. So it was continuing to use the stores of sugars and stuff from the year before to continue to work and work on the activity in the soil. This was in furrow. This was a guy that put 8 ounces of biology in the furrow, and there wasn't much difference there. There was very low biological activity and very low total activity. And this was a guy that had put the biology in furrow, the ABC. His numbers, total numbers were very high, and he's right at a 50-50. This was a field of corn this past year that went on dryland corn that went 325 bushel of the acre, no irrigation. So if we can, the reason I put that up there is it is the importance of getting the fungal working in your soil to bring those nutrients to the crop.
That field was on the program for a long time? Or?
No, it's only like the 3rd year. You know, it had good soil fertility, okay, but he's never had corn like that. The year before on another field, he had averaged 300 as well, but he's got good fertility. He watches his P's and Q's, and he did a very good job, you know, with everything. But
And he got rain.
He got rain at the right time. Yeah, he did. That's always the key thing. So.
Starter fertilizer with that?
What?
Starter fertilizer with that?
No. No starter. This guy doesn't use starter fertilizer at all. He used to broadcast, but he's got some cattle manure, and his numbers on his soil aren't exceptionally high, but yeah. So that's kind of the end here. So to put this all together, what we tried to present today is there are many aspects of what you're doing out there that are causing you headaches. The chemical aspect of it, of the chemicals you need to use out there, just the soil physics, the negatives and positives out there in your soil, and then the insecticides and the different things you have to use. But we're trying to make your soil healthy, and what we're trying to show here is some of the things we're seeing that we're starting to go that direction. And if we can do that, you're going to reduce inputs. And our goal, as Ron and I had for a goal, was to reduce your inputs by at least 50%. Now, I've talked to guys, and I got one guy down in Nebraska again, he said the more he lowered his nitrogen to a point, the more his yields went up. So I think in a lot of situations we're shooting ourselves in the foot. And we've got to remember that it's the soil and the soil biology that we've screwed up. That ratio of 50-50 is what feeds you. And that's what we're trying to get back to. And that's why we're going to do a lot of testing with Austin and his program to verify that, because I don't like BS, I like numbers. And we're getting there, probably not as fast as we'd like to for you guys' sake, but this is the path we're on, and that's how we want to serve you, and hopefully you guys will continue in that direction as well. So that is, are there any questions on any of this? I know we've covered some of this stuff before, but I'm trying to bring it all together so you understand what's happening out there in your soils and why the frustration occurs. So, yes.
Like that test right there, what was that cost? Like you just went through like 6 of them.
$10. We buy, I don't know, we've done a couple hundred of them over the winter, but we go out and we get a soil sample. And with this soil sample, we send this into Midwest, and we get a regular soil sample on it. We get a Haney test on it so we know what the carbon is and all the other nutrients in there. And then we do this test as well so we know what the bacterial to fungal ratio is, okay? What's really, this is important, but what's really important to us, what I think is really important is, so we can do all of this, but is it still, the question is it getting into the plant? And that's why we're doing the tissue testing. We spent a lot of money on this kind of testing this year to make sure that this isn't a bunch of baloney, that we're actually getting it into the plant and into the leaf. This kind of shows that it does by the yield he had on this piece of corn. But this part is very inexpensive, and you have to do it right. It's a little bit tricky. We were getting inconsistent results, and so we called the company, and they said, oh, you need to do this and this, do it this way. So, and when we started doing it the right way, then things came out very consistent. But this is very accurate, and at least gives us bacterial to fungal, and but it's very inexpensive to do. Anybody can buy this kit. It's $100,
What were you doing? What did you do different? What was wrong?
I don't know. My son was doing the testing, and he talked to him. I don't know. I'd have to have you talk to him.
Verlyn, I think it was, I remember right, Steve was saying something, you had to put like 3 drops on.
Yeah.
And you have to like wait in between, I know that was one of the things he was telling me.
Okay.
But then there was a second thing you had to ask Steve.
Yeah. I don't remember what it was either. I think so what you do is this. It's very simple. What you do is you take the soil sample, you put it in a little tube, you put a reagent in there, you let it set for 20 minutes. You take a little dropper and you take and you put it on this card like that, and you put it in the center there, and you put 3 drops on there. But we were doing it too fast, I think, like Britt said. And then you take there's a program on an app that you download on your phone. You take a picture of it, and it gives you the numbers just like this right away. And if it doesn't come out, if it doesn't come out right, it'll tell you right away, and then what you're supposed to do is you're supposed to put 3 more drops on and then do it again, and then it gives you an accurate reading. So we had to learn the tricks to it, but it's used in countries around the world to do this kind of thing, because we're not, you'll find out we're not the only crazies out here. There's going to be more and more companies that are going to be concentrating on fungal. Clint started down the fungal road before he started with us for a couple of years with a mycorrhiza product, right, Clint? Yep. And he saw the advantages of it in his cornfield. What did it would have taken 2 or 3 less passes with the irrigator where you had the fungal in-furrow?
So 3 seasons ago, I took a cornfield, and it's a pivot, it's a full circle, and we had a guy, the company that I'm using for that mycorrhizal product, they had a guy down in Kansas do it on a cotton field, soil moisture probes. So I thought, well, I'll do it this year with corn, because I sell soil moisture probes, so I have extra ones laying around, and I split the pivot, and then I put 1 probe in, and then 100 feet over, I put the other one in. Same line, same tower track, everything. And by the end of the season, like, I ran it over every time, but on your moisture probes, you get the line that shows when it gets down to where it needs more water. On the side that I added the mycorrhiza that year, it never got anywhere close. Now, the side that didn't have the mycorrhiza, that water moved through that soil so fast, it wasn't even funny. I easily could have saved 3 passes of watering. Last year, the most I watered, I think, was 6 inches of water out of a pivot. And I use moisture probes on every one of them now. But I've got guys that have used 9, 10, 11 inches last year, and I can't believe they use that much water. But yeah, there's definitely a huge notice, and that was after 1 year of using the product. That was the first time that product was used on that field. So, but it's a species mix is what I use.
Yep.
But I actually use that with the Biomentum this year in a test, and using both together was 10 bushel difference. So it's strictly mostly AMF fungal. Now the Biomentum's got a lot of different types of fungal.
Yep.
And I feel that if you can afford to use both in conjunction, that's good. But my mission now is I'm trying to go away from as much synthetics as possible. If my opinion, fungicides and all these products are band-aids, because every year you're going to have to use a fungicide. And once you start getting your fungal up to that, close to that 50-50, I feel that you're not going to need to have that disease. I talked to a guy that lives about 35 miles south of me, and he's been, he's regenerative farming, cover crops, no-till, everything. And I was talking to him last week, and he's a new potential customer. He's interested in Biomentum. But he didn't have any rust issues last year and no tar spot. And I don't know what his fungal, he's been doing it for 20 some years. So, I mean, he's built that soil up. I've seen it on soil tests, just between my field and the neighbor's field, who's been no-till. I do tillage. Their biology numbers are up. I mean, it's 100% true that tillage destroys your biology, and fungicide I feel does the same thing. But I've seen enough evidence now that I feel like working with Verlyn is going to get me on the right track. So I've cut back a lot for fertilizer. It's a slow process. It ain't a 1-year fix. But you got to be in it for the long haul. And last year was the first year I ever actually had to use fungicide on corn. I didn't want to, but I wasn't confident in the Biomentum last year because I was new to it. But Justin here's my friend that does drone spraying, and we did a trial with the Biomentum on a cornfield, split a field with Biomentum on half and fungicide on the other half. Both products stopped the rust. Tar spot moved in late August, and visual appearance on the fungicide side of the field, there was tar spot in every single plant. You could see it. I don't think it made any yield difference because it come in late enough, but on the Biomentum side, it was there, but you had to look for it. It wasn't night and day on every plant. So I feel like coming into the tar spot, that it will eventually, hopefully, help us control that. And I'm kind of going a little bit overboard. My goal is to put a half gallon Biomentum on every acre this year, because what I'm trying to do, it's different applications, but what I'm trying to do is I'm trying to bring my levels that we're finding, my fusarium and all my diseases like that are just red all the way through the roof. I'm hoping that with over application, in furrow, a couple foliar passes every year, I can bring those numbers down and hopefully control everything just by using his product, the Biomentum. So I've seen some amazing things with it. We stopped white mold and 4-foot tall beans one year, the first year we used it. So 2 passes of fungicide didn't do jack for that field. And that's when I called Verlyn and I said, hey, I can't stop this white mold from spreading. So he come out the next day and we looked at it. And the day after that, I sprayed it with, we did a half gallon of Biomentum, and I did 60 pounds pressure at 25 gallons an acre. You come back 2 weeks later, it was all gone. A lot of damage was done already, but there's nothing, not a product like it.
Did you leave a strip? Just curiosity.
Yeah, we did.
Did you? Okay. You could see the difference?
Oh yeah, we saw it. And Verlyn brought his penetrometer out, and he showed us the difference. In 2 weeks' time, we changed that soil structure. So we went back out on the sprayer, and then we went out to the edge of where we didn't spray, and Verlyn stuck it on the side that we didn't have it, and it was yellow, borderline red, and then we stuck it to the side that 5 foot into the spot where we actually sprayed it, and it never come out of the green. And we did that 3 spots in that field. When you can see that kind of results that fast, you know you're doing something. But it's one of those things where you can't just jump in on it. Find a spot, one of your tough fields, get out there, try it, play with it, because it's not the same for everybody, but for me, it's been one of my biggest things that I can see change. Of course, I'm using the tools to see the change, but it's a long road. It takes time.
Yep, but if you're going to want to see instantaneous results, then this is not the way you should go, but it takes a while to change that soil biologically, and so it's a process. Now, I'm not saying you need to do this, but where we have, the more we've put on, the faster we've changed things, just like Clint said. But we're still in the process of researching that. Different soils are going to react differently. So we're still doing a lot of testing with it. And
Verlyn?
yes.
I just want to say for the gardens, you saved my cucumber plants. I Weyhrich sprayed them with Biomentum, and they were already dying from like frog eye or a mold or something. And I had lots of cucumbers at the end of the season.
Oh, well, good.
Yeah, I mean, I knew I'd lose the whole patch once it got in there, and I,
Yeah.
like Russ said, go try it. You aren't out anything, and so I mean, it helped in the gardens too. That Biomentum, it just gave it what it needed to kick the disease.
If you're hesitant about it, put it on your garden, because you'll see a huge difference. My wife loves canning tomatoes and making salsa and all that stuff.
Yeah.
Used to plant a dozen tomato plants, we're down to 6. Because we have so many damn tomatoes, we don't know what to do with them. And I'm not kidding, this stuff.
I gotta get them out to grow first.
Use the Biomentum, I swear to God, you'll have.
Does it kill rabbits?
No.
Not really. It doesn't kill hardly anything, except diseases.
Yep.
What is the mycorrhiza product you're using with it?
It's a product called Mycomax,
Yeah.
farm fungi. New Age Farming is the name of the company.
It's a good product.
It's a very good product.
Yeah.
I had, so I have another, when I was selling it to a customer, that's kind of how I found Verlyn too, a mutual customer of ours. I didn't know he was using Verlyn's product, and I found out later. I said, well, I use some biologicals, and you want to try this one, so he stuck it out there on his field. And he says it's always nice when the neighbor pulls in and asks what the heck you put on your field, because it looked better, because dry, you know, especially on dry land, the field across the road's burning up, and you got a good field that's still green yet. You know, it makes people stop in and ask. And he still uses both. He'll use Biomentum, and he uses this Micromax in conjunction with this field. I told him, I said, we should test your field to see if you even need the Micromax anymore, but he just says, I'm just gonna use it. But yeah, I mean, the mycorrhiza plays such a huge role in your fields. And when that's usually the one that you see is low. Like, I bet if you took that test out to most of our fields, you would see, you know, really low numbers on fungal because of all the crap we're using. I'm guilty of it. I'm trying to fix my ways. So
It's a process.
it is. But I work a farm show in Norfolk, and I have a lot of gardeners come to me and ask me questions. And I had a gal do with the Micromax product, do a greenhouse experiment. She's got a little greenhouse in her backyard. And she used mine, another product called Myco Seed Treat, and then Control. And they were all individually potted tomato plants. And the Myco Seed Treat was like 2 species of mycorrhiza, and then the Micromax had 8. And she said it was very consistent. The Control was set, and then the Myco Seed Treat with only 2 species was 1.5 times the yield. She weighed everything she harvested. And then the Micromax product was 3 times. So she was like, she comes every year and gets a little, I call them, they're little, it's like I'm dealing drugs to these gardeners.
Yeah.
Because you put them in these little Ziploc blue bags, and then you go up to their house and you go, here you go, and then you get a $20 bill and you leave. So I get a little nervous sometimes when I do that, but yeah. But
A dry product or liquid?
it's
Yeah.
a dry product.
Dry.
And that particular mix is light sensitive. So when you mix it with your in-furrow starter, you want to try to use it up within 24 hours, because as it sits in the sunlight, it will deplete. So
Yeah.
it's a little bit harder to use. That's why I like Verlyn's product. It's a lot more forgiving and a lot easier to use. I feel for the cost of it, that's why I'm going 12 or half gallon for an acre, because I don't feel like $12 is a waste of money compared to what everything else I'm putting out there is. So I'm willing to put that $12 an acre out to hopefully fix what I got going on in my soil. So
We'll find out
yes.
with Austin. So he'll let us know. Next year at this time, we'll report back. Okay. So, any more questions, comments? You bet.
So I saw Howard had a deal on social media that he had an ad at the Super Bowl.
Yeah.
And the flyer or whatever. So where is that whole process at with that
Okay.
burn down?
That organic burn down product, they finally got it through EPA, at least one of them, so that they can market it now here in the US. And so that's what they're doing. I've got about 5 270-gallon totes in my warehouse that he needed a place to store. It didn't freeze. And but they're going to be marketing that heavily this year. He's on the road all the time right now. So yeah. What?
When he gets bored driving, he calls me all the time, just taking up his time.
So just get on the call.
Yeah, get on the call, does he need someone to talk to?
What's that product called?
It's Contact Organics, and they renamed it, and they call it Firehawk now. So very good product, very effective. Vineyards just love it. go down along the plants. They don't have to worry about, you know, the stuff that we showed up here, the micronutrient loss.
It improves the soil.
It does. It does. It actually increases biological activity, especially fungal activity. So very, very good product. I think they're going to do very well with it on the market.
Is it
Yeah.
getting price-wise where it
No,
put it on broad acres?
they've improved it that to the point where it's concentrated more, and so they can use smaller amount in water, so to speak. You know, instead of like, what was it?
100 gallons at one time.
Yeah, but that's going to help the price. But for an organic guy or somebody that doesn't want to use Roundup type, for a broadcast, it's going to be $50 an acre right now. Okay, but for some of those guys, they don't care.
Especially crop lighting.
More, yeah, a lot.
For now yet.
Yeah. Yep. So anyway, the Brittany included a price sheet in there. None of the prices have changed since last year, right? So if you have any questions, the program hasn't changed. The prices haven't changed. We're keeping everything the same. And if you have any questions on recommendations, contact myself or Brittany. If the program you're using is working for you, we can continue that. You understand now the direction that we're headed biologically with all this to hopefully bring your soil back into balance. You've got lots of stuff out there. You cannot be careful with nitrogen. Do not try and cold turkey that. Ron tried that one year and it bit him in the butt. The P and K, I don't know about. If you're, depends on where you're at biologically in your soil for you to start cutting back on that. If you're getting along good with what you're doing, be careful with cutting back. Go a little at a time, try it on some acres, not all of it, okay? I don't want to see any of you have a train wreck. You got to have the biology there in order to make that work.
Hey, Verlyn.
Yeah.
Just a note on that, with my soil sampling that I've been doing, that you start with the product, and then in time you'll see that your need for it after year will come down on your soil test. So I'm still spreading by soil test a little bit, so I'm just not doing the high formulas, I'm bringing it down, but
You've reduced the amount of P and K.
the need for it is reducing.
Yes.
Just naturally, like according to the soil test, what's available.
We have a colony up by Lake Benton that we've been working with for the last 3 years. I think they've been putting on a pint. My partner up there in northwest Iowa was working with them. He passed away here about 10 days ago. So, but they were telling him that their P1 levels are continuing to come up. So, without, changing fertilization or anything else. So, yes, that's what you should see. I don't have enough numbers to back that up, so that's why I don't have the guts to tell you guys, go ahead and cut your P and K out. That what Ron said to me at one point was that it would make me nothing would make me happier than not have to have these guys buy a lot of fertilizer. So that was his goal as well. But hopefully with Earth Optics, we'll get some of those answers if Austin is doing his job.
No pressure.
So, anyway, any other questions, comments? If not, thank you all for your attention today, and hopefully that some of this information was helpful, so.
Thanks for dinner too. Very good.
Thank you.
Thank you.